Hello everyone and welcome to today's video. In today's video we're going to be talking about the selfish genes and junk DNA. Now this is going to be it is actually a very wide topic that we can go on and on about. However, I'd just like to point out the headlines regarding these two things. So scientists previously believed that all nucleotides have a specific function. That means that if we have more nucleotides, then the organism must be immediately more complex, right? False. The thing about it is that this has been refuted by something called the Cvalue Paradox. I don't know if there's a video right now on the channel regarding the Cvalue Paradox, but I highly recommend reading at least about it or watching a video. It's super interesting to think about. So this refutes the idea of junk. This actually enhances the idea of junk DNA, because junk DNA, essentially these are multiple nucleotides or large segments of nucleotides that do not have a specific function. That means and refutes the idea that we have more nucleotides, more complex. Now, this concept clashes with the selectionist paradigm. What is that? Essentially, it states that most, if not all, nucleotides would be affected by selection acting at the level of the organism. Now, a breakthrough discovery was done by McClintock, first in plants and then in animals, regarding jumping genes, which are called the selfish genes, by the way. Or mobile elements. Those elements, in essence, were genetic elements that were more likely to change their position in the genome. This key concept brought to light the possibility of dynamic genomes before the emergence of modern genomic techniques. Now, this concludes our video, but I'd like to go through a conclusion real quick. And basically, it's that we have two things. Junk DNA, yes, we do have areas where we do not know their main function, however they are believed to have regulatory roles in our genome. And second, we've got the selfish genes, which is a very wide topic regarding how areas of the genome can be shifting from place and going and getting placed in a different section in the genome. Honestly, that, just like I said, is a very wide world, and we're going to be discussing a few of these concepts and more in future videos. I'd like to thank all of you guys for watching. This has been Ali from B Biology.